Hi everyone, thanks very much. I'm happy to be here to give this talk, and thanks so much to the organizers for inviting me. I hope that this is going to fit really nicely with the talk that Vijay just gave, carrying on with a bit of a genomic epidemiology theme. So in particular, I'm going to speak about some work we've been doing recently, thinking about how we can use pathogen whole genome sequences to estimate this quantity of the serial interval, and to kind of talk you through the steps that I will present today. So first I think I probably don't need to motivate for you after the last talk why we want to use genomic epidemiology, but I will think about why it is that we care about the serial interval and why we want to use pathogen genomic sequences to try and understand it. I'll then kind of take you through briefly our method that we've been working on before presenting an application to COVID-19 clusters from Victoria. And I'll say that a lot of the methods that and yes, the application to COVID-19 in Victoria was with our collaborators at the University of Melbourne and also the Victorian Department of Health. So maybe this slide goes without saying after the last talk, but when we're thinking about what genomic epidemiology is and what it's helping us learn, in general, we're trying to learn something about disease transmission from pathogen genomic sequences, in our case, whole genome sequences. What transmission is here can mean many different things. A number of them we've just seen. We can be thinking about what's the geographic distribution of our disease, what are particularly common modes or patterns of disease spread, what happens if we try and do something to combat transmission, as well as thinking about the population dynamics of the pathogen itself and what that does for the human populations that it's affecting, or animals or otherwise. And kind of a common theme that we see throughout this field is that sequence data has been proven to offer a really high resolution view of how pathogens spread, but this is imperfect in some ways because of our sampling and our sequencing strategies, and in some ways just because of the nature of randomness, really. I wanted to kind of position the work that I'm going to speak about today in the field in what I hope is a bit of a useful framing, and I borrowed this figure from this nice paper by Hill and others, which kind of reviews a lot of the current challenges within the field. And in my opinion, we can think about a lot of work that's been happening in genomic epi at these two different scales. We've got our big population scale analyses, a lot of our phylogenetic, phylodynamic analyses that are really interested in, like, what's the broad picture of how transmission is happening? Our ancestral reconstruction, where does it come from? How is it moving around an area? On the other hand, a lot of the other work has been more at this local scale down here. So where we're really interested in locally in an outbreak, what's happening, who's infecting who, what kinds of people are infecting other people. And to do this kind of analysis, we typically need like really dense sampling. We need to be seeing infector-infectee pairs, and a lot of them in general. Something that I've been trying to think about recently is what happens when we're kind of in the middle of this. And I do think that this is not a perfect framing of absolutely every piece of work. I think that a lot of phylodynamics work kind of comes into the middle here. But particularly as we're doing more routine genomic sequencing, we're starting to maybe be able to dig into the details of what's happening at a local level, but we don't quite still have that person-to-person resolution. So what can we still learn when we're in this middle regime? And as I say, one such area we've been thinking about estimation of serial intervals. I think that these are an interesting quantity to be thinking about in this middle area, because we're trying to estimate ourselves a population level quantity, which I'll describe in a second, but which is related to transmission pairs specifically. So the serial interval is described, that's jumped over, sorry, but can be defined as the length of time essentially between successive cases in a chain of transmission, or more specifically, the length of time between symptom onset and an infector and an infectee. So throughout the talk today, I'm going to talk a lot about some generic infector-infectee pair A-B. If A had symptoms at some time, B had symptoms at some time later, then we have one observation of the serial interval between them. So you can see there how it's giving us this idea of speed of transmission. Why is this important? Well, it might be obvious, but I'll tell you anyway. It's Obviously very helpful for us to know about the speed at which transmission is occurring, how fast is it spreading in a population, is it faster or slower than what we've seen before. We can use this to inform our surveillance efforts. If we want to do contact tracing, we should be knowing like what kind of times that people were in contact to inform that. And as well, the serial interval kind of cascades down into all these other quantities that we're interested in. So it's used to calculate some different reproduction numbers. So one of the classic ways of calculating R0 here from Wallinger and Lipsitch in 2006 is that R0 is approximately equal to 1 plus the exponential growth rate multiplied by the serial interval. So obviously, if we want to get an accurate estimate there, then we need to be understanding what the serial interval is. And hence the serial interval gets used in all kind of downstream epidemiological estimates that use reproduction numbers like herd immunity thresholds and so on. Now the majority, or all to my knowledge, of existing methods for estimating the serial interval do not use genomic data. They require directly observing transmission pairs, infectors and infectees. So typically we would go ahead and contact trace a bunch of pairs of cases, which we assume in most cases represent direct transmission from A to B, from C to D, and so on. This is giving us direct observations of the serial interval, and then we can parametrically fit our favorite distribution to that, or do this non-parametrically to estimate the population level serial interval distribution. When I was preparing for this talk, I wanted to look up some recent, the most recent I could find meta-analysis of serial interval estimates for original lineage SARS-CoV-2 or COVID-19. And I found this preprint by Zhu and others that was released this year, and they compiled 99 different studies to estimate the serial interval, and every single one of them used contact data. I will admit I didn't read them all, but all of the ones I looked at least were assuming these direct contact trace pairs, many of which were taken from household settings. As you imagine, it's easiest to try and contact trace people in a household setting. We know who contacted who and when. And this is kind of a common pattern in serial interval estimation. I see it as there being two kind of key challenges with this. First, getting contact data can be difficult, expensive, ethically challenging if transmission of a particular disease is criminalized, or difficult to share because it's really personal information about who contacted who and when and where. As a result, if we end up taking serial interval estimates from specific transmission settings in which it's easier to get this contact data, for example, households, if we think that transmission is occurring differently in those settings, if we think that transmission is typically faster in a household, for example, then we might be getting a biased estimate of our population level serial interval. So this motivated what we were thinking about today. And the question is, can we then use pathogen whole genome sequence data as a proxy for this contact data to estimate the serial interval? The motivation then is that we're going to be able to use this if population level kind of routine sequencing is happening. I'm going to be able to apply this in broader clusters that maybe give me a more representative estimate. As part of this, I'm going to recognize that I'm not going to be seeing all the cases, and I'm going to need to take that incomplete sampling into approach. But the main aim is that we're going to be able to get a method which is fast and gets us cluster-specific or time-specific or variant-specific estimates that we can compare in different settings to see if the serial interval is changing. Okay, so I'd like you to imagine that we have a set of different case clusters. I can get this to work. These case clusters could have been defined however I want. I've got 3 of them in my example here. They might be genomic clusters, building a phylogeny, selecting clusters on it. They might be epidemiological clusters associated with particular transmission sites. And I'm going to try and estimate the serial interval in each one of them independently. And the data that we're going to restrict ourselves to are knowing the symptom onset time, each of our cases within each cluster, and we're going to have their pathogen sequence. But critically, we're not going to know who infected whom. We're not going to use any kind of contact data to inform that. And like I said, we're going to try and use this whole genome sequence, the difference between them, as a proxy for contact data, much as we do in transmission reconstruction settings, but taking that into account that we're not going to know all the cases. So we're going to say, I'll define this more specifically in a second, that if any case A and B are clustered in the same cluster, their viruses are closely related, and they have what we're going to describe as realistic timing, then A and B are a plausible transmission pair. And one thing we are going to restrict here is that we're going to say that In fact, I'll talk about that in a second. But A and B are a plausible pair if they meet these three different measures. However, like I say, we're going to need to consider two different sources of uncertainty in this. We might not know who infected whom directly. We might have more than one plausible infector of each infectee. And even if I do find a pair, A-B, it might not be that there was direct transmission between them. I might have missed someone in an intermediate way in that kind of alignment. So our approach in brief, hopefully this is as much detail as I can fit in the time. First, we go ahead and do that I just described. Identify all plausible, possibly indirect transmission pairs in my cluster who have closely related sequences. So for this, we use a simple genomic distance measure. I say it has to be below some threshold G, which would be informed by my knowledge of the mutation rate of the virus in question or pathogen in question. We're going to say that they need to have what I'm describing as realistic timing, so the difference in their symptom onsets needs to be within some pretty generous threshold. And specifically, this is what I was about to say before, we're going to restrict to the fact that our infector has to show symptoms first. So we're not going to consider negative serial intervals here, though we do allow for pre-symptomatic transmission. So we get our set of pairs or plausible pairs that meet this criteria, and then we're going to go on to sample n plausible transmission trees given these plausible pairs. So I'm sampling an infector for each infectee to build myself up a transmission network. So looking at my little diagram over here, let's say that for my cluster 1, these are all my plausible pairs. So A is plausible with B, but not plausible with E. Then I go through this tree, and for each of the people, I sample one of the people who could have been their infector. And I actually do this according to these two different criteria here. So people who are closer in genomic distance and closer in time are more likely to be sampled. So building in that uncertainty, but using what we know about the closer together cases are more likely to represent transmission pairs to kind of inform that. Then I'm going to have this set of some number n of transmission trees, and I'm going to estimate the serial interval in each of them independently before kind of combining everything at the end to get one overall serial interval estimate. And importantly, that kind of combining at the end is going to need to take into account uncertainty both within and between the different trees that I've sampled. So I've got a whole bunch of these trees. Within each one, when I estimate the serial interval, I'll get some uncertainty, and then also the uncertainty in combining across n of them. So I included a little schematic view of the kind of method. We're up to stage 3 at the moment. This was specifically for the Victorian application that I'll speak about later. So we've got our sequences. This describes the sequencing for that Victorian analysis. We've done some clustering. As I said, that could be genomic or what I'm calling epidemiological or exposure site, like particular settings of interest. And I go ahead and sample this set of transmission networks. And now I need to go on to estimating the serial interval in each of those. And as I describe here, we're going to be doing that with a mixture model, which I'll speak a bit more about. So to take this idea of the fact that we haven't sampled everyone into account, we're going to consider that all of our inferred A-B pairs might not be direct transmission, like I said before. So we'll consider 3 different cases. The first of these is that, okay, maybe A and B were actually direct. Then what we see between A and B is a true serial interval. Great news. Maybe they were direct. It could also have been indirect. We had some unsampled cases along the way between A and B. The third one is slightly more complicated, but not too much. This is what we call co-primary infection, inspired by this paper by Vink and others in 2014. This is the case in which an unsampled person actually infected both A and B. So under our criterion before, where we look for close people, we expect that in settings like this, even though A and B did not have transmission kind of flowing between them, it's definitely possible that someone we missed infected the both of them. So we fit a mixture model that takes all of our sampled serial interval networks that takes these possible transmission pathways into account. So thinks about when we have this window between A and B, so the time between symptoms of A and B, it could be any of these 3 things in reality. And very briefly, the way we do this, so we're going to assume that the true serial interval distribution is gamma distributed, just because it's nice and flexible and also positive. So if A and B were a direct pair, then the time we see between them is one observation of the true serial interval. On the other hand, if there were unsampled people, let's say M of them in between A and B along my transmission chain, then I'm going to model this unknown M as a geometric random variable. So there was some geometric number of missing people in between them. Then what we observe between A and B is actually M plus 1 serial intervals. So if there were one missing person in here, then what I've seen is 2 serial intervals, and so on. If there were 2 missing people, I've seen 3 serial intervals. And it turns out we can model this nicely with a compound geometric gamma distribution. So this is a geometrically distributed number of gammas added together. The last case for coprimary is slightly more complicated again. Hopefully you can believe me, I can talk a bit about it a little bit, that the observed interval we see between A and B ends up being what can be described as a gamma difference distribution. So it's telling us that there was a gamma serial interval between question mark and A, there was another gamma serial interval between question mark and B, and what I see is the difference between them in the length of time between A and B. Happy to talk about that a bit more if people want. So the result of this is we get a mixture model with 4 different parameters to fit. 2 of them describe our serial interval. So I didn't say, but here I'm using the mean and the standard deviation of the gamma instead of a shape and rate or anything like that. So I've got my mu and my sigma, my mean and standard deviation of the serial interval to estimate. And as part of this, I also get 2 kind of additional parameters. 1 pi here that comes out of the sampling rate, essentially the rate at which these geometric missing people occur. And the last one is basically describes the proportion of infections which are coprimary. And that gives us, well, a slightly informally written distribution for the time that we see between A and B here. If you're asking why does the direct case not come into this, I've just got my compound geometric and my gamma difference. It's because you can think about the direct case being simplification of the compound geometric when M is 0. So these kind of collapse into one. And so we go ahead and do the serial interval estimation, like I said, in each of our trees and then combining them together. There's a lot of steps of combining things. And we use maximum a posteriori estimation to fit our data to this model, involving using some beta priors on these 2 parameters, W and pi. Basically, these represent some kind of knowledge of the sampling level in our population. At what rate are we missing people? And so that's why we chose to put priors on these. So for my last, before I get to our data application, to try and convince you a little bit more that this actually works, I've just presented some results here on a simulated outbreak. So we used the outbreak simulation function in the Outbreaker R package to simulate whole genome sequences for an influenza-like outbreak, which was also simulated, the outbreak. So we simulated with R not equal to 2 and a serial interval with mean 4.5 days to get a kind of true known outbreak with whole genome sequences for every single case. And then we applied our serial interval estimation method to that simulated outbreak. But actually looking at 10 different experiments in which we increasingly downsample our data. So I pretend I don't know, well, in the one case up here, I put the fully sampled outbreak into our method, and then I downsample it all the way down to only having 10% of cases randomly selected to see how this thing works. And I've just shown you the results here for the serial interval mean and standard deviation. So the dashed lines here are the true simulated values. My interpretation of this is that in all scenarios, the estimates, at least the means shown by the points here, are pretty good. However, the uncertainty really starts to blow up when we are not seeing a high proportion of the cases. I would say that around about 30% is the cutoff where I think this is not really working anymore. So I'm taking from this that as long as we think we've got about 30% of the cases being sequenced, this is going to be good. Above that, we might start to be concerned about the estimates that we were getting, or at least the uncertainty around them. So with our collaborators in Melbourne, we applied our kind of methods to the first 2 waves of COVID-19. Yeah, thanks. To the first 2 waves of COVID-19 in Victoria, and I've just shown for you here, we picked 10 clusters from each of those first 2 waves to have a look at, and I've plotted them out here. So we're having about, 20 to 150 cases in these 10 different clusters, and we're estimating the serial interval in each independently. As I described at the top here, the first wave here, these are genomically defined clusters. We built a phylogeny, used the cluster picker tool to select them off. And in the second wave, these are epidemiologically defined clusters. I don't normally include this, but for this audience, I thought I'd include the phylogenies of the two different waves to try and convince you a little bit more about why I think that decision made sense. In Victoria, the first wave of COVID-19 was very, I guess, categorized by local sporadic outbreaks, and we see that reflected on the phylogeny. These, like, I don't know, very distinct clusters along it, so they're colored by the cluster that we picked. Whereas on the second wave, they had a lot more localized transmission, so we still see some nice separated clusters, but we see this horrible mess down here of really short branch lengths, really similar clusters, a lot of identical sequences, and we found that genomic clustering just wasn't really cutting it for us on this. This would all be one big cluster under pretty much every criterion that we tried, and so that's why we decided to pick these cluster sites of interest. So we are using a little tiny bit of contact data here just to identify cases that were associated with outbreaks in particular schools or workplaces or long-term care facilities. So here's our estimates of the serial interval given those 20 different clusters, sorry. So I've just focused here on the serial interval mean. So again, these are mean and 95 confidence interval estimates of the mean serial interval. We can see that they're all pretty closely in line with that 4.8 days that the meta-analysis earlier got. We see a little bit of variation between them, not too much. We did a statistical test of whether any of these serial intervals were significantly different, and after we took like a multiple testing correction into account, indeed, none of these were significantly different. But, well, I'll speak a bit more about that in a second. But we do see pretty wide uncertainty in some of these estimates arising from that large uncertainty in who infected whom. Which I think is natural, given the fact that in this data, we think we've got about 50% sequencing. So they had strong genomic sequencing, but we're still missing half of cases on average. Where we did see a little bit more significant difference between our clusters was when we had a look at these different kind of exposure site settings that they were seeing outbreaks in the second wave. So here I've expanded out to look at 94 different clusters of COVID-19, most of which can be categorized into these different types. So aged care, healthcare, housing, schools, packing and meat processing plants, and then a bunch of ones in gray here that didn't really fit nicely into one category. And what we did find is that when we compared the serial interval in these different settings, indeed, actually, meat packing and processing plants did, even after a multiple testing correction, have significantly shorter serial intervals than the others. So we think that transmission is happening more rapidly in this setting, which at least with my knowledge in British Columbia of COVID-19 outbreaks, kind of follows what we saw in practice. I don't know. You'd have to ask someone who was from Victoria how their knowledge about it. But yes, we did find that one to be significantly shorter, particularly in comparison to these healthcare facilities here, where we were seeing these confidence intervals completely non-overlapping. Given the time, I will maybe skip over this and just say that actually, the confidence intervals don't show on the board, but I will say that serial intervals go into these downstream estimates of the basic reproduction number, and we did find in some settings that using a cluster-specific serial interval as compared to a kind of standard literature estimated from a different population serial interval could be pretty impactful, up to about 3 times a difference, our estimates of RT. So I'll finish up just by saying, yeah, well, in my opinion, I think it's nice to think about this intermediate regime where, with this data, I don't think we could have done transmission reconstruction. Packages like Outbreaker and Transphylo that are meant to infer who infected who. We have really low diversity sequences, a lot of identical sequences, and a lot of missing cases, particularly in wave 2. But hopefully we were able to show that we can still learn things about transmission, even given that. Here we're estimating serial intervals without needing contact studies. And if we are able to get more of this kind of broad population sequencing, it's going to be easier to kind of compare and contrast different intervals across time and space and setting. And what I'm kind of thinking about for this next is, could I do better than this if I combined genomic and contact data? I do have it for these Victorian intervals, so maybe the best thing is to use everything I've got. And are there other kind of quantities that we can think about that would be applicable in this kind of modeling framework? So with that being said, I'll leave it there. I've included a link to our paper on this that came out last month. And I'll say thank you to our funders from Australia and Canada, and thanks to everyone for listening.
My question is, so do you, when you reconstruct the chain, do you need the intra-host variation information?
Inter-host variation. Yeah, we do not use it, just because they are routinely sequenced one sequence from each host in general. If we think that there is significant within-host variation and that essentially the sequence that we sequenced was not necessarily the one that was transmitted, it would be beneficial. Most of the data settings I've worked in, we don't have that information, so it has not really been on the board to think about.
Yeah, another question is, so when the people are really crowded and so the transmission is really fast, so do you compare your model, the prediction results with the real epidemiology, the real contact information?
Or as in, have I compared
I mean, the people, the record people to people contact with the inferred contact.
I see. So we have done a little bit of that in that maybe I included the slide here. We compared, yes. So we essentially did this whole method in the way I described, and also did the method only using contact data. And these are kind of comparable pairs of clusters, so like the blue and the green and the red and so on. I have not looked at how the inferred transmission networks we get compare to what the contact data tells us, in part because we have so much uncertainty in that, but it would be interesting to look at. But yes, we did do this comparison.
Okay, thank you.
Yeah.
Thank you for a great talk. Could you show me once again the slide, our approach in brief?
Oh, yeah. Yes.
So, yeah, I just wanted to understand the workflow correctly. And so regarding the second point, so you sample plausible transmission trees from, so basically the first step creates the network, right? So directed graph shown here.
Yes, the first step would be to get this network.
Yeah, yes, the network. And so the second step is about extract tree inside network, right? And so as down here, as shown down here.
Yeah, this third one.
Yeah, so graph theoretically speaking, so you create, choose n subtree inside network, right? And so is it arbitrarily choose?
We're preferentially sampling according to these two rules here. So if B, where's a good example? D here is like possibly infected by both C and B, and I would draw it randomly, but with higher probability. If, let's say, C had closer distance and closer time, I would be more likely to select C more often than B.
Okay,
it's like a probability weighting we apply.
Okay, so then each tree has probability or likelihood, or right?
Yes, yeah.
Variability.
We don't use that right now, but we've been thinking about it.
You don't use it?
No.
Okay, so the sampling is not very at random, not random sampling. So no, but according to some.
Yeah, according to this probability weighting. So we use the probability weighting to sample them, but then after that we treat all of our entries as equally likely. We could do a weighting. I kind of didn't want to, like, these rules are somewhat arbitrary, and I didn't want to use them like too much, which is why we chose that. Yeah.
So serial interval is defined for each edge, right?
Yeah.
And so the third step is estimating serial interval distribution for each tree. And so, so it is like a histogram, like discrete probability. I mean, so because serial interval is about each edge, of that tree. So what exactly that does it mean?
Yeah. So we treat each of these edges, you're right, is one observation from the serial interval distribution. And then for each tree, I have, in this case, 1, 2, 3, 5 observations of it that I'm fitting to that mixture model, wherever that was. Yeah. I'm treating that as 5 observations from this mixture model distribution.
Okay, so I understand. Thank you.
I think also my question is related to this slide. So eventually in the network, if you sample the tree, so every edge will be in a tree. So probably I don't know. Maybe you don't need to sample the tree. I think the weight is more important. Which infection is more reliable? I think that's important.
Yeah, so I've thought about this a bit. It is not the case that we always sample every unique tree. I've checked that basically, given this because the size of our clusters are way bigger than this little example here. So, and we often have a lot of plausible infectors for each infectee. So every edge maybe ends up in the tree, but every unique combination of trees does not occur, which is why we chose to do the sampling. But we are thinking about future applications of this, which basically take those weights and calculate things on them directly.
I think if you assign the weight to each edge, you calculate maximum weight spanning trees.
Yeah.
More interesting to me.
Yeah, we've been thinking about that for essentially looking at like if different groups of people are infecting more than others, like vaccinated, non-vaccinated, or different age groups. And then, yeah, you can just look at these weights directly.
One question again on this slide. In practice with the SARS-CoV-2, which distance do you use for the genomic distance and the time? Because I assume that in a small cluster, it's quite likely that all the genomes are the same. Or
yeah,
that's the question.
In the Victorian analysis, we used essentially one SNP close to it for the genomic threshold, and the time one we set as actually way more lax. So we wanted the genomic information to be more informative than the serial intervals, so as not to kind of bias ourselves to short ones. So I think we use symptom onset time less than about 20 days or something very generous to incorporate that we might be missing cases. But yeah, genomic distance, we really restricted ourselves to very close clusters and pairs. And one thing I didn't speak about is that it's highly possible, like for this case G here, that we don't find any plausible pairs for a person if there's nobody close, and then we just say we don't have enough information to work with G, and we discard them from that analysis.
Thank you very much.
Thank you.